and welcome to the Ideas Forum. My name is Erzsébet Szatmári. I am Assistant Professor of Neuroscience at East Carolina University in the Department of Physical Therapy. And it is my great honor to introduce today my collaborator, my colleague and great friend from ECU, Dr. Alexandro Di Donna from Brody School of Medicine, Department of Anatomy and Cell Biology. Alexandro is a neuroimmunologist, neurogeneticist, studies, well, for me, you are the best neurogeneticist and neuroimmunologist at ECU. Alexandro studies multiple sclerosis, neuroinflammation associated with multiple sclerosis. And today he's going to talk about a new story, Ataxin-1, as a modulator in multiple sclerosis. Alexandro, welcome.
Thank you, Erzsébet, for the nice introduction, and thank you everybody for having me here. So today I'll be talking about this project that actually I started during my postdoc, and now is one of the main projects in my lab here at ECU. Just one single slide about me. I'm a molecular biologist by training. I was exposed to neuroscience research during my PhD, and then I started working on multiple sclerosis during my postdoc at UCSF. And then now at ECU, I run my lab working on the connection between neurodegeneration and neuroinflammation in chronic neurological diseases. So the way I prepared this talk, I wanted to give a brief introduction on multiple sclerosis, so we are all on the same page. For those of you who are not familiar with this disease. So MS is a relatively common autoimmune disease affecting the central nervous system and is the first cause of non-traumatic brain injury among young adults, which are defined as people between the 20 and 40 years of age. The main disease phenotype of MS is CNS inflammation and the demyelination. In other words, we observe the breakdown of myelin sheets that wrap the axons. We also observe extensive gliosis and eventually axonal injury and neurodegeneration. These cause a broad spectrum of symptoms, both cognitive and motor symptoms. The cause of MS is still unknown. So what I'm presenting, here I'm presenting two of the most common models that we have right now to explain how multiple sclerosis starts. On the top of this slide, we have the so-called outside-in model, in which we postulate that immune response in the immune cells in the periphery gets activated and then migrates into the CNS, where they start attacking the myelin and causing myelin breakdown. On the other hand, there is this other model called inside out, where we postulate that the disease starts in the brain, in the CNS, with a neurodegenerative event that causes the death of neuronal cells and the spread of epitopes that eventually recruit immune cells. So the immune response in this model is seen as a secondary event to the neurodegeneration. We still don't know which one is true. We have data supporting both. It could be possible that the disease is also a combination of both processes. But so far, we still don't know what is the epitope that triggers the autoimmune response against the myelin. So some information on the epidemiology of this disease. We know that it's much more common in Europeans and European descendant populations. And there is also a female-to-male ratio of 3:1. And this ratio has been increasing during the last century. Again, the age of onset is between 20 and 40 years of age, but we also have a rare case of pediatric MS. And we also observe a strong influence of latitude and ancestry on risk. And there is also a certain component of family history in about 20% of MS cases. The typical disease course, the clinical evolution of the disease, can be divided in two main phases. We observe at the initial stage that is called relapsing remitting, in which we observe these peaks of clinical disability. They are followed by a complete or incomplete recovery. This initial stage can last for several years before the patient enters into a second stage that is called secondary progressive, in which we observe the accumulation of clinical disability. So the first stage is mostly inflammatory. The secondary progressive stage is mostly neurodegenerative. A small portion of MS patients, about 10-15%, they enter directly into the progressive phase, and they are called primary progressive patients. So as I mentioned, we don't know yet the etiology and what triggers the disease, but we know that the MS has a genetic component. This is mostly due to studies on twins, in which we observe a much higher concordance among twins compared to the non-twin siblings and the general population. So if we plot, for example, the lifetime risk of developing the disease against the genetic identity, we observe that monozygotic twins that share 100% of genetics, they have a much higher risk, about between 30 and 40%, compared to the general population that is less than 5%. But the evidence that even in twins there is not a perfect concordance should tell us that genetics is not the only factor in this disease. In fact, MS belongs to the so-called complex disease group, in which we observe a polygenic inheritance. In other words, we observe many allelic variants that are common in the population, each one contributing a small portion of the overall risk of developing MS. And we also observe a strong influence of environmental factors. In fact, right now, what we consider the most up-to-date disease model for MS is a multifactorial disease in which we observe environment, genetics, and epigenetics, all of them concurring to the overall MS risk. So these diseases, because it's not as a Mendelian diseases, the classic genetic tools, they are not really effective in studying these complex diseases. A big change in the field of genetic research in MS has been introduced when this tool called GWAS was first developed. So GWAS, they stand for Genome-Wide Association Study, and these are alternative paradigm to classic Mendelian genetics, in which we can increase the power of genetic screening by testing large group, big cohorts of patients and cases, genetically unrelated, for the uneven distribution of specific genetic markers along the genome. The most common markers are single nucleotide polymorphisms, and we test for the association with a specific trait, in this case, MS risk. This allows to identify specific regions of the genome that are strongly associated with the disease. So after more than a decade of GWAS screening in MS, we now have a pretty clear picture of the genetic of MS. And as the fundamental contribution was done by the creation of this big consortium that is called IMSGC, and this is the current members of the consortium, in which labs all around the world were started sharing expertise and samples to reach a really high power. And this is from 2019. This represented the up-to-date genetic MS genetic atlas by testing a cohort of MS patients close to 50,000 individuals. The IMSGC was able to identify the more than 200 genetic variants strongly associated with MS. In particular, all of these variants can explain up to half of the genetic risk of MS. What is still missing is probably constituted by smaller effects that are still not detectable with this sample size and possible genetic interaction between genes. For this talk, I will be talking about one of these regions that was recently associated with MS risk on chromosome 6, in which the peak of association falls within a gene that encodes for a protein, a Ataxin-1 protein, that has been known for being associated to another neurodegenerative diseases that is called SCA1, or spinocerebellar ataxia type 1. One thing that is crucial for this talk is to understand that GWAS is a statistical tool. So all these associations are purely statistical, but GWAS doesn't say anything about the biology behind this association. So a lot of effort has been put forward to understand what are the molecular mechanisms underlying these associations. And also it's important to remember that because we observe an extensive linkage disequilibrium in a human genome, identifying the true causative variant in a genetic locus is not a trivial effort. So a lot of tools have been developed to understand which are the true causative variants in these GWAS associated regions. And in particular, our collaborator at UCSF developed this tool to bioinformatically screen all the variants within each locus and use publicly available information from the ENCODE project to associate a specific score for each gene in the locus to the risk of to the probability to that specific SNP or single nucleotide polymorphism to be the true causative variants. And in this case, when we tested this method for this locus, Ataxin-1 was the top hit, the top gene in all the different cell types that we tested, both in the immune system and in this the central nervous system. So this evidence represented the basis for our further follow-up in vivo. So just a little bit of background about Ataxin-1 and this other disease that was previously known as the main disease associated with Ataxin-1 dysfunction. So Ataxin-1 is a polyglutamine protein that is expressed in many tissues in the body. Belongs to the polyglutamine family of protein, because in the N-terminus there is this polyglutamine stretch that in some cases get pathologically elongated, and this causes a toxic protein build-up in specific neuronal population, in particular the Purkinje neurons in the cerebellum, that cause motor symptoms. There is a really good mouse model to mimic the human disease that I present here in this slide. And we observe the extensive death of Purkinje neurons in mice that have this polyglutamine stretch artificially elongated from the wild type 2 glutamine in mouse to 154. the mice lose weight, they get motor symptoms, and at some point also some cognitive impairments. So this was caused by the aggregation of ataxin-1 in the central nervous system. But very little is known about the possible pathogenic function of ataxin-1 in other tissues, in particular in the immune system. So what we did was to take an in vivo approach to try to understand how and why ataxin-1 could be associated with a higher MS risk. To do so, we use a very good and widespread, very well characterized model of MS that is called experimental autoimmune encephalomyelitis. This is an artificial disease that can be induced in many animal species, mostly rodents, by immunizing the animals with short peptides from myelin proteins, such as MOG, MBP, PLP, together with specific adjuvants that are able to boost the autoimmune response against the myelin components. So after immunization, typically the animals start developing clinical symptoms around day 10 after immunizations, then reaching a peak of disease within 1 month before entering a chronic phase of the disease. These mice develop a flaccid paralysis, extensive demyelination, and neuroinflammation. By playing with the animal mouse strains and the specific peptides that can be used to immunize these animals, we can also model different disease courses, such as relapsing, remitting, or progressive course. So in this case, what we did, we obtained from a collaborator an ataxin-1 knockout mouse model that doesn't show any gross phenotype, and we induced the EAE in these animals. And to our surprise, we observed that knockout mice deficient of ataxin-1, that is the top curve, develop a much more severe disease compared to the wild-type controls. We also immunize heterozygous animals carrying one copy of the gene, and those animals develop a disease that was in between the wild type and the knockout, possibly indicating a gene dosage effect. And these different disease courses were also recapitulated by the mortality rates of these animals that reached close to 80% in the knockout animals, and they stayed less than 5% in the wild-type controls. So we could conclude that ataxin-1 exerts a protective effect on the autoimmune response. Next, we were curious to understand if this polyglutamine stretch was also implicated in this beneficial activity of ataxin-1. So we employed that model that was developed for the in the SCA1 field, and we induced the disease in these animals. But to our surprise, we couldn't observe any major effect in the transgenic animals compared to the wild-type controls. Probably indicating that this polyglutamine stretch is not involved in this immunomodulatory function of ataxin-1 in EAE. So, as I mentioned before, this gene is expressed in many tissues of the organisms, and these knockout mice that we used were global knockout. So at this point we still didn't know which was the main cell target of this novel immunomodulatory function. So we started looking into different models and possibilities, and we were driven by this observation that in wild-type animals, after inducing the EAE, we observe an increase in the expression of ataxin-1. The mouse has two isoforms that we called 1.1 and 0.2. Both of them were increased in the spleen of these animals. So we decided to look more in detail in the function of Ataxin-1 in the immune system. So to isolate the effect of Ataxin-1 deficiency in the immune system, we employed a variant of the EAE paradigm that is called adoptive transfer. In other words, we can induce EAE pathology by transferring immune cells from an EAE immunized mouse to a naive recipient. So when we transferred knockout cells, Ataxin knockout cells in wild-type animals, we observe exacerbated disease course compared to controls receiving wild-type cells, mimicking the observation in the global knockout. So we could conclude that the efficiency of Ataxin-1 in the immune system is per se sufficient to recapitulate the phenotype we observe in the global knockout. So as we know, in the spleen, so we transfer basically splenocytes. We have multiple cell types. And we know that EAE is mostly driven by pro-inflammatory T cells. So we could envision two possible scenarios at this point. In one scenario, we could see a direct effect of Ataxin-1 on CD4 T cells, or in alternative, we could envision an undirected non-cell autonomous effect through the APC function, the antigen presentation. So to discriminate between these two possible scenarios, we look at the level of pro-inflammatory cells that are defined as Th1, Th17, that produce interferon gamma or IL-17, and also the Tregs that are, on the other hand, anti-inflammatory cells in splenocytes from knockout and wild-type mice that were stimulated with the MOG peptide that we use for the immunizations. In this first set of experiments, we observed that the knockout cells showed an increased level of Th1 pro-inflammatory cells. But when we purified the CD4 T cells from this complex cell mixture and we repeated the experiment, we couldn't observe any significant difference between genotypes. So we could conclude that the ablation of Ataxin-1 is probably mediates an increase in pro-inflammatory Th1 cells to a non-cell autonomous effect, possibly through increased antigen presentation. So to test this possibility, we look at the main antigen presenting cells in the spleen. So we look at monocytes, B cells, and also we look at CD4 and CD8 as controls. And we observe that by testing using flow cytometry, two main markers of cell activation, CD44 and CD80, that are expressed on the surface of immune cells, knockout B cells showed an increased activation in knockout mice compared to the wild-type controls. So we decided to focus on this specific cell compartment that we know that works as a professional antigen-presenting cell type. And at the functional level, we could show that the increased activation of these cells were able to drive the proliferation of T cells in co-culture experiment in panel G. So the knockout B cells could drive the maximum proliferation of T cells at lower level of MOG peptide compared to the wild-type cells. So at this point, we had enough element to hypothesize that probably Ataxin-1 works through the B cell compartment. So we decided to look in detail and carry out a very extensive characterization of the B cell compartment in Ataxin-1 knockout mice. Here I'm presenting an overview of the different B-cell subsets that are present in the adult mouse spleen, each one defined by a different combination of surface marker. So we employed flow cytometry to look at all these different B-cell subpopulation in the spleen of wild-type and knockout mice. And we observed that a specific subset of B cells, in particular the B1, A and B, and the marginal zone B2, were differentially expanded in the Ataxin 1 knockout mice compared to wild types. Other T cell subsets, such as the B-regs and the follicular B2, were unaffected. And this was we repeated this in naive mice and also in EA immunized mice. As an additional control, we also look at different T cell subsets, such as naive T cells, memory and effector T cells. We could not identify any difference between wild type and knockout, confirming that these effects are probably mediated through the B cell compartment. In fact, when we also look at the B cell and T cell infiltration in the CNS of EAE mice at disease onset, we observe a statistically significant increase in knockout B cell infiltration compared to wild type, but we couldn't observe any difference between the T cell infiltration, confirming that the ablation of Ataxin 1 mostly affects the B cell function. So we then decided to complement this histopathological analysis by looking at some specific B cell function. First, we look at the immunoglobulin production. We know that B cells produce antibodies, and we could test their level in the serum. So we tested the total IgG titers. In particular, we tested IgG and IgM, two of the 5 classes of immunoglobulins, and we observed that the knockout mice have higher IgG, naive mice, and also after immunization. After immunization, we also observe higher IgM. We also develop an assay, ELISA assay, to test only for the anti-MOG, so this peptide that we use for immunize the mice, and we observe an increase in anti-MOG IgG level in the knockout mice. So we could conclude that Ataxin 1 negatively controls the IgG production. Then we also wanted to look at the specific pathways that could have been dysregulated by Ataxin 1 deficiency. So we employed RNA-seq to look at different genetic signatures between knockout and wild-type B cells at baseline and after MOG immunization. And we observed that at baseline we found ontologies related to cell activation, confirming our flow cytometry screening that we did before. And also after MOG immunization, we observed many ontologies related to cell cycle and cell division. In particular, we could confirm the upregulation of key cyclin genes, in particular the D1 and D2, in the Ataxin knockout B cells. Another interesting observation was that most of these genes that are upregulated in knockout B cells, they also physically interact as part of a large network that is involved in cellular mitosis. So it's also so we went back to the literature and we observed that this link between Ataxin 1 and cell proliferation was already present in the literature in other fields, in particular in the cancer field and also in the same SCA1 field. And in particular, they identified specific signaling pathways in the cells that mediated this Ataxin 1 effect. So we were curious to understand if the same signaling pathways took place also in our story. To do so, we employed a variant of a classic flow cytometry that is called phosphoflow to look at the level of phosphoprotein in the cytosol of different immune cells. And we could confirm that in knockout B cells, there were increased level of MAP kinases and STAT proteins. They are some of the main signaling pathways in immune cells, and in particular STAT1, that is upregulated in B cells, has been implicated in Th1 differentiation. That was what we observed in our in vitro experiments. So from the SCA1 field, we also know that at the molecular level, the role of Ataxin-1 works together with other proteins in a big molecular complex that sits on the promoter of specific genes and negatively modulates their transcription. In particular, there is a paralog of Ataxin-1 that is called Ataxin-1-like that works together with Ataxin-1 to stabilize this downstream effector that is called Capiqua, CIC. So to understand if this same molecular complex is mediating this effect in B cells in the context of EAE pathology, we perform conditional knockout studies. In particular, we were able to obtain these conditional K-LOX lines to delete Ataxin-1-like and Capiqua in T cells and B cells using specific K driver, such as the CD2 and the CD19. And then we produce these four conditional knockout lines, and we induce EAE in all of them. What we observe on top of this slide, we could observe that when we deleted Capiqua specifically in knockout in B cells, we could partially recapitulate the global knockout phenotype. But we could not observe any difference in the Ataxin-1-like conditional knockout in B cells. No effect was found in both T cell lines. That also tell us that probably the T cell compartment is not affected by Ataxin-1 deficiency. So we could put together this model in which Ataxin-1 in B cells regulate the antigen-presenting function that eventually drives the polarization of CD4 plus T cells to a pro-inflammatory phenotype. So all the data I've presented so far have been collected in mice. But what about MS patients? So we were lucky that in the lab there was another postdoc and that she was conducting some epigenetic screenings in a cohort of MS patients. In particular, here we are seeing some data on DNA methylation at MS onset. What we observed that at the disease onset, B cells experience a very strong hypomethylation in their DNA. And among the genes that were mostly demethylated, there was Ataxin-1. It is in the top 10 genes. In particular, we could identify 4 methylated regions that are hypomethylated in MS patients at disease onset. And we could confirm that these regions overlap with functional elements that regulate the expression of Ataxin-1 in these cells. And also at the experimental level, we could confirm the hypomethylation controls the expression of Ataxin-1 in B cells that is actually upregulated in MS patients. So we could put together a story in which the upregulation possibly works as a feedback loop to negatively regulate the immune response by negatively control the B cell function. So this is so far all the data we have collected, and in the short, these are some future plans that we are currently working on in the lab. We want to further dissect the role of Ataxin-1 in B cells at the level of nuclear structure and chromatin structure. And in the future, we also want to look at Ataxin-1 in other autoimmune diseases, possibly for developing therapies that can modulate the autoimmune response. So finally, I have a lot of people to acknowledge that made this possible. My lab at ECU, John, Christy, Alex, Bryce, and Cindy, then my old lab at UCSF, where I started this project, and also all the collaborators. Huda Zogby is the scientist the first discovered Ataxin-1 working at Baylor College. Her postdoc Q-Min Tan that shared with us some these conditional knockout lines, and then Nikos Patsopoulos at Harvard University that was involved in the initial GWAS screening. And finally, I would like to thank all these sponsors that funded this project: NIH, National MS Society, and Italian MS Society. Thank you for your attention. I would be happy to take your questions.
Miles, Brian here from UNC. Really, really great talk. That was awesome. I kind of had two questions for you. One being a more epidemiological question. That being, I'm not sure if anybody's looked at this. Has anybody seen increased comorbidity between SCA1 patients and MS patients?
Not that I'm aware of. The issue here is SCA1. So MS is much more common than SCA1.
Sure.
SCA1 is considered like a rare disease. So I believe that also that makes it more difficult to have a robust data about possible comorbidities.
Okay, great. No, that makes sense. And the second question is a little bit more mechanistic. So it looks like a lot of your data is driven through these Ataxin knockout mice or Ataxin knockout cells,
Yes.
where you're losing function, basically loss of function. And so I'm wondering, have you thought about or have you tried overexpressing Ataxin-1 either in mice or in B cells to see if that can potentially rescue any of the MS-like phenotypes?
That's a good question. There is no overexpressing Ataxin-1 model, so we could not do that in vivo. We are thinking about using lentiviral vectors to try in vitro on B cells and see if this could modulate and rescue the phenotype. Yeah, so most of the field is dominated by SCA1 tools. They work they are interested in the polyglutamine stretch. So there are very few mouse lines looking at the physiological function. I'm currently developing a conditional knockout Ataxin-1 model to start looking at other cell types. So here, all these lines were obtained through collaborators. So yeah, there is no Ataxin-1 overexpressing line.
Gotcha. Well, just coincidentally, this isn't something that we use regularly, but we do have an Ataxin-1, I think a 2Q and an 82Q lentivirus. If you're ever interested in potentially using that, we have plenty in our lab, so just feel free to reach out.
Thank you.
All right, great talk. Thank you.
Alexandro, I was wondering what you think about this theory of Vartanian. I think I might have brought this up before, for sure, to John. He has this beautiful paper in a Journal of Clinical Investigation showing that Clostridium perfringens would be the cause of MS. Do you believe in this hypothesis, or what do you think that MS might be caused?
Yeah, so when I was still at UCSF, they started a big screening about the microbiome in MS, and they found a few bacterial species that are associated, they are more prevalent in MS patients. And when you transplant these species into the mouse gut, they are also able to modulate the EAE phenotype. So if they are the real cause, I'm not so sure. Could be a consequence of the disease or a modulator, but it's a really hot topic. Another evidence is that EBV, so this Epstein-Barr virus infection, that is a virus of the B cells, has been associated with a higher prevalence incidence of MS. So now people are studying the involvement of EBV in B cell function as a possible trigger through molecular mimicry. We don't have solid data because this paper came out a couple of years ago. Now we are start seeing the first evidence of possible viral protein that are similar to the myelin protein that could trigger an autoimmune response. So this is a really exciting field. Yet I don't personally believe that the microbiome can be the cause of MS, but maybe could affect the severity of the disease.
When you look at the infectious agents that are involved in MS, is there any overlap with those that were shown to be involved in Alzheimer's? Because Duke-UNC has this idea, right, that Alzheimer's disease is...
So I know of a study where they compared multiple diseases. There is a genetic overlap with specific other autoimmune diseases for sure. And there is a certain overlap also with other neurodegenerative diseases. I don't remember exactly the numbers with Alzheimer, but we know that in Alzheimer, if you do a GWAS, so GWAS in Alzheimer picked some immune genes. So we know that now in Alzheimer, there is an immune genetic component. So I will not be surprised if there is a neuroinflammation also driving Alzheimer's disease. But I am not sure about, I don't remember by heart the numbers of this overlap. I'm sure that, for example, lupus, there is an overlap between lupus and MS at the genetic level. There is another one in the bowel disease. I'm not totally, some type of cancer, but I'm not totally sure about Alzheimer.